Hi. So we left off with talking about Darwin and his theory of natural selection and evolution and how we got to that point through understanding natural selection by artificial selection and just all of the cool ways that we have that can support this hypothesis, this theory now over the periods of time. So today we're going to dig into it a little bit more because natural selection isn't the only way that populations can be affected and changed. And so we're going to go through various kinds of what's called microevolution or evolution on a small scale. This is generally between generations, so we can see changes accumulating within generational time periods. So for our objectives today, hello, click. Okay. We're going to define microevolution. We're going to talk about kinds of natural selection. We're going to talk about a little bit of math. Okay. I promise it's not scary math, I promise. And then we're going to talk about some of the different ways that populations can have pressures and changes occurring over time. So let's talk about microevolution to begin with. Microevolution are small changes within a population from generation to generation. So this is something that you could see if you were someone who planted flowers every year, or what Mendel saw with his pea plants, right? Mendel's peas, he was able to look at changes in a population from generation to generation. If he started seeing things where he had a mutation that resulted in peas having two different colors, for instance, within the same pea flower, that would have been a mutation that could have resulted in the population changing after generation to generation. This is because of those heritable traits, those genes that are involved. Okay, so if we were to look at, say, some generations of beetles, and we have a first generation where we have 75% of the population having the green allele and 25% of the population having the brown allele. In this instance, green is recessive to brown because we see that individuals who are heterozygous have the brown phenotype. Okay, we know this from learning about genetics. So that is the dominant gene. But in this case, it's not as common in the population. The population actually has more individuals with the recessive green alleles than we would see in that. So when we count up the genotypic frequencies or the percentage of those genes within a population, my first generation has 75% of the individuals with green alleles, or 75% of the alleles are green, and 25% of the alleles are brown. But if I look at my second generation, I have a shift in that change where I'm going to see 71% of the alleles now being green and 29% being brown. This means that I am seeing small changes within the gene frequencies from one generation to the next. And this is what the micro evolution is. We see this by that genetic reshuffling through sexual reproduction. We see it through potential changes from mutations, but this is what we're talking about. Okay, so if we look at and remember, natural selection occurs involves a couple of things. Okay, first, variation has to exist within the population to begin with. If all individuals in the population are genetically identical because they go through asexual reproduction and there's no physical variation, there's no genetic variation, we're not necessarily going to see natural selection. Now, that doesn't mean that we won't see mutations arise in those individuals and we won't see variation occurring as a result of that. But if it's just everything is the same, we're not going to see natural selection. Okay, so natural selection requires that variation. Members have to be different from one another. In addition, any of that variation we see has to be inherited because of those genetic differences. It's not that individuals paint themselves different colors or that I dyed my hair purple and so now I have variation and I have purple hair and you have, brown hair. That isn't what it is. It has to be something that's genetically acquired, okay? And the last caveat here is that natural selection has to increase fitness. Natural selection has to result in individuals who are better adapted, surviving and having more babies. They're going to have a larger proportion of offspring, which means they have higher fitness, and those offspring are likely to survive and have their own higher fitness. And so we will see that that trait becomes more common in the population as a result of this natural selection. So I have an image here of fitness, right? And we're talking about these green and brown beetles. Well, if green beetles just naturally have a gene that results in only producing two offspring per individual, they are going to have lower fitness than the brown beetle who has a gene to produce 4 offspring per individual. And so we see that there can be changes in just even the fitness numbers, who has the most offspring. It can be because of survival, it can be because of predation or competition or a whole lot of other things, anything that might act as a sort of pressure. So one of the other things I need you to remember is that natural selection is not perfect. Adaptations are not perfect. And natural selection doesn't look through genes and go, ooh, I don't like that gene combination. Okay, natural selection is acting on phenotypes. And phenotypes can be at a cellular level. It can be the enzymes being produced. It can be things that we don't see, right? But it is going to be a phenotypic change as a result of that protein synthesis from that genotype. Okay, evolution is going to be constrained by the variations. You're not going to suddenly wake up and find a beaver with a chainsaw hand because that's the best way to cut down trees. Okay, instead, what we see is beavers actually have teeth that grow forever, and that is going to allow them to eat through that hard, dense wood in order to knock those trees down to build their dams. Okay, beavers don't actually eat the trees themselves, they eat the bark on the young limbs and the leaves. Okay, populations are going to have genotypic variations, especially if populations are undergoing any sort of sexual reproduction because of meiosis and that genetic recombination we see, as well as then that haploid gamete going with another haploid gamete randomly to produce a new individual. So all the things that we've been learning about really tie into this understanding of natural selection and evolution. And when we think about what we've learned, this should make sense, because these are the things that we're saying, okay, well, we saw this, and this is why this is the way that it is. And we do this a lot without even thinking about it. I know we talked about, I talked about in this class, Fat Bear Week. Fat Bear Week is fantastic. And if you aren't aware of it, what it is there's bears in Katmai National Forest, National Park, that gorge themselves on salmon and gain a ton of weight. Well, if you happen to have a genetic trait that allows you to put fat on more as a bear, that might allow you to survive better through the winter through hibernation and give you an edge the next season when it comes to getting out of hibernation and finding food and mates. And so that could be something genetic. And during Fat Bear Week, we actually see that offspring from certain individuals do really well and become competitors. And a lot of that has to do with that genetic variation and that genetic variation being passed on to their offspring. Okay, so when we talk about natural selection, it's not also always just like one phenotype is better, or it varies. So there are actually 3 different kinds that we see of natural selection. So these traits generally in a population have a range, and we consider them a bell-shaped curve. If you remember back to genetics, and we were talking about like human height and those polygenic inheritances, and we end up with like an average height, okay? Natural selection tends to fall within that, where you will have individuals that have extreme phenotypic traits, but the majority of individuals are going to be in between those phenotypic traits in a naturally occurring population. Now, when we have pressures, those pressures are acting on those phenotypic ranges, and different kinds of pressures on different phenotypes result in different changes in the populations. Okay, so let's talk about it in a way that's going to make sense. I have this image here that shows the stabilizing selection, where my individuals who have the extreme phenotypes are selected against. I have my directional selection, where one extreme phenotype is selected for and the other and the average phenotype are selected against. Or I have a situation where my phenotype in the average, that middle phenotype, is selected against, and both extremes are selected for. So I have examples. Ready? Stabilizing selection. Okay, stabilizing selection is when the average or intermediate traits in a population are the ones that survive the best. This is what we see most often in most situations. Extreme phenotypes are going to stand out in their environment. They're going to potentially be negative, and the mice are a fantastic example. Mice generally do come between gray to black coat colors, and we're going to have a range of those colors in between. So you can imagine that I have a white mouse, a gray mouse, and a black mouse, but I can have shades of colors in between those. If I'm in a natural environment, like here in Jacksonville, North Carolina, black mice are going to stand out because they might be in the green grasses and things. And if you are a predator, that black mouse might be easier to see. And so the black mice or the dark colored mice can get selected against. They get consumed and potentially before they even reproduce. So that's going to reduce their fitness. And if those genes are removed, right, they're not passed on to babies, then those genes aren't in the population anymore. Okay. White colored mice, also not going to do so great here because it is very green and brown, and so those whites are going to stand out and they are going to be consumed fairly quickly. As such, we tend to see that gray colored mice camouflage best because they're in between both of the extremes and they survive and reproduce. And so we see a shift from the original population of having this kind of broad, wide bell curve to more of a narrowed bell curve where the middle phenotype is the most selected for. We actually see this happening in mice, but we see it in things like human baby weight. Okay, if we think about a human baby and you try to picture in your brain the average sized baby, my bet is that you are telling me a poundage between 7 and 9 pounds. Okay, between 7 and 9 pounds, if someone tells you that they had an 8-pound baby, you're like, perfect. That is a good sized baby. That's neither really, really large or really, really small. But if someone comes to you and they tell you that they had a 2-pound baby, that's a little baby. That's a tiny baby. That's preemie that probably needs to be on NICU and other care. Or if someone comes and tells you they had a 15-pound baby, you're going to ask what the heck you were eating during the pregnancy because that is a very large baby. Well, today's medical technology allows us to potentially have those babies in those extremes, but imagine that you are a peasant woman in Ireland in the 1300s. Okay? At this point in time, you're in your village. You probably have other women helping you give birth, maybe a midwife if you're lucky, but it's not like today. Okay? If you have a 2-pound baby, generally speaking, there are complications that are resulting in that baby being born prematurely. That baby is not going to be fully developed, especially in its lungs, and that's why NICU can be very, very essential for people who have premature offspring. That is going to be a thing that happens, but if you're a 1300s, like, the year 1300 peasant, there is no way that you are going to be able to help that baby survive, okay? In addition, there's a really good chance that the mom is at high risk for something and then the mom might die. So that trait is going to be removed from the population pretty quickly because individual humans that are having babies that small of a size aren't going to regularly survive, okay? And the same thing if they have a 15-pound baby. C-sections weren't really common at that point in time. In fact, I don't think there might be historical records of C-sections, but it wasn't really a common practice. So you can imagine if there's a 15-pound baby and you cannot pass that baby through the birth canal, that individual is probably going to die and the baby is going to die. And so we see this kind of range selection of human babies being between 6 and 8 pounds or 6 and 9 pounds, because that's what our bodies can handle, relatively speaking. We see this in birds as well. A bird might be able to successfully lay 11 eggs, but if it can't raise 11 offspring, then laying those eggs is a waste of energy. And so we see that, like, if you go to look at a robin's nest, it's an average of 4 eggs. If you go to look at a bluebird's nest, it's an average of 5 eggs because that's what they can successfully raise. And so we see clutch sizes as a response to that as well. If they have extremes on either end, it's not good for their fitness and therefore not good for adding their genes to the population. Okay? Directional selection is a little bit different. So directional selection is when one extreme phenotype is favored and another is not. And I love that everyone talks about giraffes in terms of feeding, but this is the best image I could find that kind of matched the other ones and helped us make sense of this and have like a solid comparison. Giraffes don't have long necks for food. Giraffes have long necks for making babies, okay? They use their necks as a lever to compete for mates. Now, finding food can be great, but if you go out and see, a lot of studies have shown that giraffes actually feed at about the same level as everyone else within the canopy of trees. Anyway, okay? In this instance, though, if I had a giraffe with a short neck, okay, that giraffe is not going to be able to compete for resources, whether those resources are foods or mates, and it's going to then potentially die. That's going to be selected against. And individuals within a certain range of that neck height are not going to do very well. But if we have long-necked individuals who are going to be successful in competing for those resources, we will see that trait beginning to become more popular in the population over time. And this is something that we saw in the pepper moths. That's a very classic example of natural selection. Human height. This is something that we see whenever an extreme is selected for because the environment has changed. Okay. And then you have disruptive selection. This one is a little bit different because in this case, the extremes are selected for. And what that ends up meaning is that if I have an environment where I have a population of the normal bell curve range, and I start having changes in my environment where some of my environment is now very dark and some of my environment is now very light, my individuals who are light and dark are going to survive really well in those environments, but my gray individuals are going to stand out. And we do see this in things like the moths in pollution-free or polluted environments. So those peppered moths, we actually saw a really big shift towards dark-colored moths during the Industrial Revolution. And we've seen a shift back towards the more average or lighter moth phenotype as a result of cleaning up that pollution. But we're seeing in some urban creatures that if you are in a park environment that gets a lot of sun, you might be a lighter colored individual as compared to individuals living in more overgrown areas or closer to buildings where it's more shaded. Some of those individuals are darker in color, and this is something that's been studied in things like snails, that because again, if you stand out in your environment, you are going to be at risk, and that's going to result in these different selection processes. Okay, so here's your understanding on natural selection, the 3 different kinds of the selection factors. I would suggest writing yourself an example, researching it, or coming up with your own example. Okay, once you're good to go, move on to the next slide. All right. At the base of everything, we talked about mutations with protein synthesis, and really without mutations, none of these other things happen, okay? Mutations are really the driving force of evolution because it's the ultimate source for new alleles. You don't have new alleles without a change in the genetic information, and that's going to be because DNA is not good at copying itself. And so we're going to have variation. Now, remember, not all mutations are good or bad. Some mutations are just there. For instance, heterochromia, where individuals have 2 different colored eyes, that's not necessarily increasing their survival or decreasing their survival and therefore affecting their fitness. It just is. It's just a mutation. Freckles are another example, okay? But when we see some changes, like a mutation that results in sickle cell or a mutation that results in a darker colored mouse in a new dark environment, that's going to be huge. Or mutations that result in natural selection pressures with antibiotic resistance. I'm going to post a video for that one. Please make sure you watch that. It's a quick video from Harvard Medical. If you haven't watched it, go watch it. It's fantastic, and it illustrates this. Without mutations, without those things, we don't see natural selection. We don't see any of these other factors. But at the end of the day, you know, mutations are fairly well understood and straightforward. Once we have an understanding of mutations, we can also look at some of the other ways that populations can change over time through forms of microevolution. One is an example like gene flow. Okay, gene flow actually kind of reduces evolution because gene flow mixes up the gene pool. This is also called gene migration because when you move, you take your traits with you. And actually, humans are a fantastic example of this. Okay, we live in a very, very global world now. You can go out and find a partner in another part of the globe who doesn't have the same ethnic race backgrounds as you, and that's wonderful. Okay, we as humans are keeping our gene pools mixed up all of the time. This can then increase the variation in the population and reduces the chances of evolution happening, where we're going to have speciation occur as a result. So when we talk about things like gene flow, it's something like this western deer population and the eastern deer population. The western deer population are a lighter color, the eastern deer population are a darker color, but if they keep mixing up, then we're going to see eastern deer with lighter colored coats and western deer with darker colored coats. So it can prevent some of these speciations that we'll see in macroevolution in our next chapter. So gene flow kind of decreases the chances of evolution, okay? But then we also have, this is a kind of natural selection, okay, but I like this one. I love talking about sexual selection. I think it's fascinating. Sexual selection is the selection of a mate according to genotype or phenotype, and this is not a chance. And generally speaking, it's going to be phenotypic, okay? You have a trait, even as humans, we have traits that we prefer, right? We have physical characteristics that we prefer. Now, individuals tend to mate with individuals of the same phenotype. And this is especially true in things like birds. Turkeys are a great example of this, okay? Turkeys are a lekking species, and that means that the males display and the females choose. What's really cool though is if you've ever seen turkeys and peacocks like on the same farm or in a zoo or something, turkeys and peacocks will often display for one another. But the females are like, no, you have nothing that I want. You don't match my phenotype. You're not the kind of male I want. And that's going to be really important at preventing hybridization. So that's a good thing, okay? Sexual selection it favors the characteristics that increase the likelihood of obtaining mates. So I had mentioned with giraffes, they actually have long necks because the male with the longest neck can fight successfully against the other males, and then he mates with the majority of the females. That's passing that gene on then to the offspring. Okay, so large antlers in deer, they use those antlers to fight one another, and deer are harem breeders, meaning that one male will mate with multiple females, and as a result, we actually do see that if males have larger antlers, their children, their male offspring, will likely have those larger antlers as well. And so that's going to put pressure onto that trait to be possible and present. The same thing with turkeys with their big fans and their bright necks and their beards, all of that is away. But with turkeys, it's female choice. Okay, so females are going to look around and they're going to choose the male that they think is best displaying the traits that they have interest in. In either case, in nature, mating is not chosen. Okay, it's not by, or as I was going to say, mating is not by chance. Mating is non-random. It is not that they just walk down the road and are like, you know what? You're the one I'm having babies with. Instead, there really is usually some sort of choice pressure because you need to decide who is going to be the best potential mix for your offspring to increase survival chances. And as a female, eggs are expensive. Eggs are incredibly expensive to make. And so mating with the best male is going to be the best choice. And so we do see sexual selection showing up quite a bit in most animal populations. Okay? The last kind of microevolution is genetic drift. So genetic drift is really cool because genetic drift is the opposite of natural selection. Natural selection is things by chance. Genetic drift is random. Okay, everyone has equal chances of survival or death. So this is really going to be very drastically noticed in small populations because if you only have 10 individuals to start with and you remove 5 of the individuals from that population, you've removed half of the population. Those genes, those alleles that those individuals had are gone. And so there is a chance that those alleles might be lost forever and never return. Okay, this is something that we're going to see happening in 2 different ways. There's the bottleneck effect and the founder effect. Okay, the bottleneck effect is what we see a lot with endangered species. And what ends up happening is that species suffer a near extinction and only a few survivors go on to reproduce. It's called the bottleneck effect because if I had a bottle full of colored beads and I poured some out randomly, I am not going to have the same number of alleles and I am not going to have the same kinds of alleles. Every single time I might even get different ones. And so we're going to find, and you're going to do a lab related to this as well, which will be helpful, that this is completely random, okay? I also really like to use, if you were in a seated class, okay, I'm sorry that you're not, I have a candy bucket on exam days, okay? I have a population of candy here and toys, and if I randomly pull out a handful, right, This handful is not the same size or type as this handful. Okay, that is the bottleneck effect. That is randomness. That is genetic drift. Okay, cheetahs are something that's really interesting in this because cheetahs have actually gone through two bottleneck effects in their species history. The first was after the last ice age, and the population reduced to about 10,000 individuals. And then again, more recently, due to poaching and hunting processes, they actually reduced really low. I don't remember what the number was, but it's very, very small. So we're actually seeing increased chances of infertility due to inbreeding in cheetahs. And we know that cheetahs are a female, as a cat, want to mate with multiple males in order to increase the genetic diversity of offspring. Female cats tend to mate with multiple males. They can't do that now. There aren't enough males to do that. And so offspring tend to be very closely related to one another, which also increases chances of inbreeding, and it increases chances of not having disease resistance. So the bottleneck effect can be really problematic, and it is of major concern with endangered species management. Okay? The other kind of effect we have is called the founder effect. And again, this would be like if I grabbed my candy bucket and I pulled some individuals out, and this was my new starting population. Okay, these are not the same as everything I had to begin with. And when I move to a new environment, not all of these are going to survive. In fact, I pulled out, this is great, I pulled out 5 little toys and 3 pieces of candy. We can imagine that the 3 pieces of candy get eaten, right, in the new population. The toys will survive. Hopefully, you're not eating toys. This is now my founder population. I'm going to have the genes of these individuals, but these genes from these candies are gone forever. So we see this in things like Darwin talking about the iguanas, right? The iguanas on the Galapagos Island are different than the iguanas on the mainland because some individuals were moved out to the island through a hurricane and they either survived or they died. And those that survived are going to have some of the factors and adaptations that allowed them to survive and result in a new species because natural selection happened after a founder effect event. They're not black and white. Okay, these are not going to only happen alone. If we have a population that's continually getting new genes in through gene flow, we wouldn't necessarily see the founder effect. So islands are usually a great place to watch the founder effect happen, but Topsail Island off the coast of North Carolina isn't because it's not hard for organisms to move back and forth from the mainland to the island. It's not far enough away. And so we'd see too much gene flow there to see the founder effect followed by natural selection. So hopefully that makes sense. If you watch any nature documentary, these things are happening and you can look for some of those characteristics that have been noticed in those documentaries. I think it's fantastic. Okay? So here's your check your understanding. Okay, I want you to look through this. I want you to make sure that you can give examples for each of these and give good definitions. Again, flashcards are your best friend. And then after you're done with that, let's talk about this in a more realistic mathematical standpoint. Okay. We can look at evolution as humans and put math to it. Okay, there's a meme out there. I love it. It's like a little bunny, and someone's like, "Biology, you'll come study plants and animals," and it's like a lady with a knife behind her back, and she's trying to get the bunny to come to her, and it goes math and chemistry. It is, but when we can add values to things, it becomes something that we can interpret and then we can understand from a genetic context within evolution. Okay, so we know that there is a gene pool, and the various alleles for all the individuals in the population make up the gene pool. We describe the gene pool by genotype and allele frequency. Okay, so we can look at genotypic frequencies and we can try to identify and calculate allele frequencies of a population. And this can help us then understand the microevolution that's occurring between one generation and another. So these peppered moths are a fantastic example. This is the peppered moth on a light colored tree. There's a light colored moth that is camouflaged and a dark colored moth. If you're a bird, you're going to end up eating the dark colored moth. Okay. In the other picture, if there wasn't a flash, you probably wouldn't see the dark colored moth as well. But if you were a bird, again, that light colored moth is going to stand out and you're going to be able to eat that light colored moth potentially before that has its offspring and reduces its fitness. So if we look at these, we can determine which trait is dominant, and we can identify the alleles, okay? And then we can determine genotypic frequencies using a Punnett square. So we're pulling some of the stuff that we learned in genetics into evolution. So what does that mean? Well, G.H. Hardy and W. Weinberg created equations to calculate these frequencies. They did not work together. In fact, they came up with it kind of on their own, and it's now become what's called the Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium is a way for us to look at a gene pool and say, look, I think I have an understanding of what's going on here, and if I can assign some values, I can make assessments and estimates about a population. So I can determine that there is a dominant and a recessive allele in a population, correct? I feel like we all understand that makes sense. And if I know that an individual has a dominant allele, they will show the dominant phenotype, which Mendel showed us we could figure out in the 1860s. I can calculate by counting up the number of individuals of a sample size that have the dominant allele, right? I can look at, say I'm looking at flowers. I love flowers as an example for this. I have 100 flowers in my hand, and I know that red is dominant to white. So if I have red and white flowers, you could count up the number of red flowers and divide that by 100 to determine the frequency of the dominant allele in the population. So if, for instance, I had 80 red flowers out of my 100 flowers, my dominant allele frequency would be 0.8 because 80 divided by 100 is 0.8, okay? Knowing that, I can do some basic math, and 100 minus 80 is 20, so I know that I have 20 white flowers. I could have counted the white flowers, I could count the red flowers, it doesn't matter. In that case, I can determine the recessive allele frequency. But if you remember, right, the allele frequency, just because I have a dominant trait does not mean that I am homozygous dominant, right? It means that I'm homozygous dominant or heterozygous. And so when I say that I have the dominant allele, it just means that one of my two alleles is dominant, and that's why I'm displaying that phenotypic characteristic. So there are two equations we actually use for Hardy-Weinberg. P plus Q equals 1 is going to be what I use to determine the allele frequency in the population, P being dominant and Q being recessive, okay? We can think about it a little bit as the phenotype as well, because if we're talking about basic Mendelian inheritance, you have a dominant phenotype and a recessive phenotype. And then if I look at those and I want to determine what the genotypic frequency is, I have to do P squared because that's my homozygous dominant individual, PQ because that's my heterozygous individual, and Q squared because that's my homozygous recessive individual. But if I do a Punnett square of heterozygotes, I actually get two heterozygous individuals. And so that's where we get the P squared plus 2PQ plus Q squared equals 1. So what does this mean in the real world? Well, Hardy-Weinberg makes some assumptions, and we have to kind of roll with those assumptions, okay? This is basically called a null hypothesis, meaning that we don't expect this to be fulfilled in naturally occurring populations. But because we know microevolution is occurring basically all of the time. Hardy-Weinberg's assumptions state that microevolution is not occurring if there's no natural selection. Well, we know natural selection is occurring all the time, right? No mutations. Again, we know that mutations happen. No migration or movement of individuals. No genetic drift, meaning there's a really large population and they're not having any sort of bottleneck or founder effect happening. And random mating. Okay, again, that would be like going down the street and picking somebody and being like, yep, we're getting married and having kids. It's not really how it works, even in nature. If any of these are going to be violated, then populations are not in equilibrium and and evolution is occurring, okay? So if we see these being violated, which we do, we can then do a little bit of fun math. All right, in the Hardy-Weinberg equation, okay? If we look at our moths from our example, and I know that in moths, the dark color is dominant and the light color is recessive, okay? I know that if I counted up 100 moths, and of those 100 moths, 80 of them were the light colored moths and 20 of them were the dominant colored moths. I know then, right, that those traits are inherited. But because dominant individuals can be homozygous dominant or heterozygous, I need to go a little bit further. If I were to cross an individual who is heterozygous, we would cross the two alleles. And so we basically do a Punnett square and we say, okay, cool. 0.2% of the eggs are going to have the dominant allele if they're heterozygous. 0.8 are going to have the recessive allele if they're heterozygous, because that's the frequency within the population. Okay, and then I cross that with another individual. So in order to figure out my p-square, I say it is capital D, that dominant gene times the dominant gene, or Q times Q, Q squared, is going to give me, in this case, 0.04, because 0.2 times 0.2 is 0.04. Okay. Then I would look at my next, and I have two individuals who potentially can be heterozygous by doing that Punnett square cross like we did with genetics. And so I'm going to say that it's 0.8 times 0.2, which is 0.16, okay? And 0.16 times 2 is 0.32. So about 32% of the genotypes are expected to be heterozygous. And again, then if I do 0.8 times 0.8 for my homozygous recessive individuals, that's 0.64, or I would expect 64% of the genotype to be homozygous recessive, and I would expect 4% to be homozygous dominant, okay? If I look at this in the next generation and I get different numbers, say in my next generation, I actually count and I get 0.3 as the dominant allele, and therefore 0.7 as my recessive allele, my numbers don't stay the same. If I have 0.3, it'd be 0.09, right? And then 0.3 times 0.7 is 0.21. 0.21 times 2 is 0.42. And then 0.7 times 0.7 is 0.49. So I would end up having a next generation, a second generation, that would be 9% homozygous dominant, right? 42% heterozygous, and 49% homozygous recessive. In that change, the population has evolved from one generation to the next. And if we see that consistently over time, we will see that these frequencies change, and we're going to see microevolution occurring. Okay? You will have a very basic question on Hardy-Weinberg on your test. It's going to have a very, very, very basic question, such as what is on your Check Your Understanding here, question number 3. Make sure you know the two equations for Hardy-Weinberg, and make sure that you can solve for the value using P of 0.3 or fill in the blank. It would be the math you can do in your head, but you can use a calculator on the test. If you have any questions, please let me know. And join me for macroevolution next, or speciation events. Thank you.